Kate, while you do that, I have a few words for you. So today marks 4 years, 4 months, 3 weeks, and 5 days since one monumental jump of paradigm-shifting spaceflight science occurred when you, for the first time, sequenced DNA beyond the bounds of Earth. Since that time, 6 of your crew members have advanced that work and demonstrated significant advancements in microbiology through culture-dependent and culture-independent identification of microbes, human diagnostics when native RNA was prepped and directly sequenced on station, and in fundamental biology when sequencing was used to assess DNA repair following cellular transformation and CRISPR-based DNA damage, all on board. And that came from the minds of a team of high school students who won Genes in Space. Your work revolutionized space science, and today marks another significant achievement with the collection, preparation, and sequencing of highly complex and multiplex samples. On behalf of the biomolecule sequencer team, aka Manon team, myself, Sarah Stahl-Rommel, Aaron Burton, and Kristen John, and the whole spaceflight science community, we thank you for continuing to push space science through your work on board and through all you do to socialize and emphasize the importance of molecular biology in space. Thank you, Kate.
Oh my gosh, you guys are going to make me cry. This is so cool. I'm just incredibly excited to see this, and I've been talking about multiplexing for years, so this is amazing that you guys have been able to do this, and it's really, really, really fun to be at the pointy end of this spear here and to get a chance to do the sequencing in space. So I'm very excited about this, and I can't wait until we're multiplexing 96 well plates and swabbing everything on the space station.
That's where we're headed next.
Subscribe for more space.